4.2 Mechanisms of enzyme action. Explain the mechanisms of enzyme action based on induced feed model. But before that, we look into the structure of enzyme. So as mentioned in 4.1, enzyme have active site. So active site is the specific site ataupun cleft ataupun groove on the enzyme where specific substrate can bind to. So all enzyme must have active site. So other than active site, some enzyme also have allosteric site. So what is allosteric site? So it's another site on the enzyme that molecules that is not substrate can bind to, either to activate or inhibit the enzyme. So awak kena boleh bezakan mana satu active site, mana satu allosteric site. So the main difference is yang kita boleh nampak dalam gambar ni, active site must be larger compared to allosteric site. So itu main differences antara active site dan juga allosteric site. So itu yang pertama. Yang kedua, active site tadi kita cakap substrate yang akan bind. But for allosteric site, something else, other molecules yang akan bind that is not substrate. So itu perbezaan yang kedua dari segi function of the site. Next, from our discussion in 4.1, we know that enzyme can speed up the rate of metabolic reaction by lowering activation energy. Okay, so what we don't know is how. How does enzyme lowers the activation energy, hence causes the rate of metabolic reaction to speed up. So the first one is by forming enzyme substrate complex. So basically referring to the binding of substrate to the active site of the enzyme. So this is the structure of the enzyme substrate complex. By forming ESC, which is the transition stage, it will bring reactants ataupun substrate close together in the correct orientation. So for a chemical reaction to occur, substrate tersebut ataupun reactant tersebut must collide in the correct orientation. So formation of ESC will allow this point to occur. Okay, so ESC will allow the reactant to be close together and then in the correct orientation. So itu point nombor 2 ni. So next, the enzyme will facilitate breaking of all bonds and then formation of new bonds, which then leads to formation of products. So ini apa yang berlaku yang menyebabkan lowering of the activation energy that will result in the speeding up of the chemical reaction. Semuanya because of formation of the enzyme substrate complex. So kompleks ni yang sangat penting untuk menyebabkan tiga benda ni untuk berlaku. So dekat sinilah transition stage yang dimasukkan tadi apabila ada formation of enzyme substrate complex. So when there is ESC form, the activation energy for that reaction is lower compared to the chemical reaction without enzyme yang ada higher activation energy. Semuanya because dia tak ada ESC form. Okay, next, baru kita discuss the mechanism of enzyme action using induced feed model. So, in the induced feed model, the mechanism of enzyme action explain the active site is not exactly complementary to the shape ataupun structure of the substrate. So, dekat sini dikata not exactly complementary. Bukannya bermaksud not complementary. So, do not miss the word exactly. Kamu tak boleh simply cakap active site is not complementary. Itu salah. Kalau dia not complementary, maksudnya the shape of the enzyme and substrate are totally different. Okay, so dalam induced feed model, kita kata active site is not exactly. Maksudnya they are still complementary, cuma not exactly complementary with each other. So here we have an enzyme with its active site. So kat sini dia punya active site. And then this is the substrate for the enzyme. Kat sini kita nampak substrate ini punya structure. Tengok structure dia lebih kurang macam structure of the enzyme. Lebih kurang sama. Maksudnya mereka still complementary. The substrate and the enzyme are still complementary with each other. Kenapa saya kata mereka still complementary? Because the substrate can still bind to the active site of the enzyme. Okay? So kalau kamu kata dia tadi not complementary, structure of substrate itu memang totally different. Meaning that the substrate cannot bind to the active site. Okay? So for the induced fit model, the active site is still complementary, cuma it is not exactly complementary. Kasi kita nampak dia punya structure of the substrate macam ni kan. Tetapi for the enzyme dia ada groove-groove macam ni. Dia not exactly complementary, but they are still technically complementary. So itu konsep yang penting dalam induced fit model. So word exactly ni wajib ada when you define the induced fit model. So in induced fit model, the active site and the substrate are not exactly complementary with each other. Next, binding of substrate to the active site will induce slight change in the structure of the active site. So here we can see a substrate and its enzyme, and we can also see that the structure of the substrate is not exactly complementary with the active site of the enzyme. Once the substrate binds to the active site of the enzyme, this binding will induce slight change in the structure of the active site. Now we can see that the structure of the active site has changed slightly to fully become complementary to the structure of the substrate. So nampak tak? Before binding, structure dia tak fully complementary. After binding, and then baru kita nampak dia become complementary. Okay, become exactly complementary with each other. So yang berubah adalah the active site yang akan berubah. Siapa yang menyebabkan active site itu berubah? Binding of the substrate. So do not say that the substrate changes its structure. No, not the substrate. It's the active site of the enzyme that undergoes slight change in their structure to become complementary with the substrate. So ini adalah point yang penting bila kita nak describe the induced fit model. So the enzyme become fully fit with the substrate because it is induced by the binding of the substrate to the active site. So itu nama dia induced fit model. So as I mentioned earlier, the active site will become fully complementary with the substrate and then allow formation of the enzyme substrate complex. Once the enzyme substrate complex has formed, it will allow formation of products, and then the products will be released from the active site. The enzyme will return to its original shape and then free to bind with another substrate to catalyze another reaction. So apabila produk sudah form, produk itu will be released from the active site. So once the product are released, the enzyme active site will change back to its original structure. So this enzyme can now bind with another substrate to catalyze another reaction. So that's it for the induced fit model. Next is 4.3 cofactors. So there are two main learning objectives for 4.3. First is define cofactors. Second, state the three types of cofactors, which includes metal ions, coenzymes, and prosthetic group. For each of these cofactors, you have been able to give at least one example for each of the types of cofactors. So what is cofactors? So it is a non-protein component required for some enzymes to function properly. So, kat sini kita ada an enzyme, lepas tu a substrate. So, if you look very carefully, the structure of the active site at this point is not fully complementary. So, when it is not complementary, the substrate cannot bind to the active site. Okay, so binding kena occur. So kesannya substrate itu akan detach from the active site. So this is why we need a cofactor, which is a non-protein component, okay, required by the enzyme to function properly. So what cofactor does is it will bind to the active site. So now we have a cofactor already bind to the active site of the enzyme. So the binding of the cofactor to the active site of enzyme will allow the substrate to become complementary with the active site of the enzyme. So tengok balik structure of the active site. Now baru dia complementary with the structure of the substrate. So, nampak tak the function of the cofactor required. So, without cofactor tadi, substrate cannot bind. Tapi with cofactor, substrate now can bind to the active site. So, itu dia kata non-protein components diperlukan for the enzyme to function properly. So, without cofactor, enzyme reaction cannot occur. So, substrate can now bind to the active site and then formation of the enzyme substrate complex followed by formation of products. Okay, next, the enzyme without the cofactor ataupun dalam bentuk inactive, we call them as apoenzyme. So bila ada cofactor, for example coenzyme ataupun any other cofactor lah, so binding of the cofactor to the active site of the enzyme will activate the enzyme. So in active form, dalam bentuk aktif, the enzyme is called holoenzyme. So once it is become holoenzyme, then the substrate can bind to the active site because it is now complementary with the structure of the substrate. So technically kita boleh anggap cofactor ni sebagai activator for the enzyme. So without cofactor, the enzyme is inactive ataupun apoenzyme. With the cofactor, the enzyme is now active, that we call as holoenzyme. So next, you must be able to state and give example for the three main types of cofactor. So the first one is metal ions. So what is metal ions? They are inorganic substances, for example magnesium in hexokinase enzyme. So nanti awak belajar hexokinase ni dalam chapter cellular respiration. So at this point awak hanya perlu boleh state magnesium 2+, an example of metal ion cofactor. The second type of cofactor is prosthetic group. So it's a group of cofactors that are tightly bound to the enzyme and are difficult to remove. So kalau prosthetic group, they can be organic or inorganic substances. Kalau tadi metal ion, dia hanyalah inorganic substances. Tapi kalau prosthetic group, it can be both, either organic or inorganic substances, tak kisah. So an example yang awak kena boleh state in the exam is heme group. So sebenarnya awak dah belajar dah pasal heme group, iaitu structure yang berada dekat dalam hemoglobin yang kita jumpa dekat dalam red blood cell. So dia akan bind with the oxygen. So the third type is coenzyme, which is the organic. So bind loosely to the active site of the enzyme. Kalau prosthetic group tadi, dia akan bind tightly. Tapi kalau coenzyme ni, dia akan bind loosely to the active site to facilitate substrate recognition or removing products. They are made up of vitamins. An example is NAD+, iaitu a type of redox agent in cellular respiration process. So ni pun kita akan belajar in detail in the next chapter. So itu sahaja untuk the types of cofactor. Lastly, 4.4 is inhibitors. So there are two learning objectives. The first one is to define inhibitors. Secondly is to state the two types of inhibitors which are competitive inhibitors and non-competitive inhibitors. So what is inhibitors? So inhibitors are any molecules that bind to the enzyme and decrease its activity. So boleh anggap dia sebagai the opposite of cofactors. So kalau cofactors tadi adalah yang akan tolong to activate the enzyme. Tetapi kalau inhibitor ni adalah the opposite. Dia akan bind to the enzyme, still bind to the enzyme, cuma function dia is to decrease the activity of the enzymes. So how? How does an inhibitor can prevent enzyme from performing its function? So by preventing formation of enzyme substrate complex. So ingat balik macam mana enzyme boleh buat kerja adalah dengan form enzyme substrate complex. Okay, so to inhibit an enzyme activity, dia kena boleh prevent formation of enzyme substrate complex. So there are two types, as I mentioned earlier, either competitive or non-competitive inhibitor. So next we will discuss the differences between the competitive and non-competitive inhibitor. So the first one is competitive inhibitor. So what is competitive inhibitor? So it's a molecule with a very similar structure to the substrate. So itu key information that you have to be able to state to describe competitive inhibitor. So the molecule have a very similar structure to the substrate. Therefore, it is also able to bind to the active site of the enzyme. So in this example, so substrate A is the substrate for this enzyme, but there is also an inhibitor with similar structure as substrate. So structure inhibitor ni kita nampak lebih kurang macam a portion of the structure for the substrate A. So because of the similarity in the structure, this is why this inhibitor can also bind to the active site. Walaupun dia tak bind kepada keseluruhan active site, tapi oleh kerana satu portion tu sama, it can cause disruption to the binding of substrate A to the active site. So as a result, the inhibitor will compete with the substrate for the same active site since they have similar structure. So because of this competition, it will lead to less formation of enzyme substrate complex. So whenever the inhibitor able to bind to the active site, it will prevent the substrate from binding to the active site. When substrate cannot bind, less ESC. So when less enzyme substrate complex form, this is why we say that inhibitor will reduce the activity of enzyme reaction. Okay, next, even though the inhibitor can bind to the active site of the enzyme, the binding of inhibitor does not change the structure of the active site. So they just bind sahaja, it will not affect the structure of the active site. So the structure of active site still maintain like the original structure. Okay, so ni penting untuk competitive inhibitor kerana if the inhibitor is removed from the active site of the enzyme, the substrate can still bind to the active site. So kalau inhibitor tu kena remove, the substrate can still bind to the active site and then form ESC, and then the product will be formed and released. So kasih kita nampak kesan competitive inhibitor adalah tidak permanent. Maksudnya dia hanya affect enzyme activity apabila dia bind kepada active site. Kalau dia tidak bind, enzyme itu can still bind to its substrate and perform a chemical reaction. So next is non-competitive inhibitor. So opposite kepada competitive, the non-competitive inhibitor molecule that bind to the enzyme allosteric site. So as I mentioned earlier, enzyme have two sites, either active site or allosteric site. So for the non-competitive inhibitor, it will bind to the allosteric site. So ni adalah original structure dia, inhibitor, substrate dan juga the enzyme. So once the inhibitor bind to the allosteric site of the enzyme, it will cause conformation change to the structure of the active site. So kasi kita nampak active site pun dah berubah. And then the enzyme pun also sudah berubah compared to yang tadi original dia macam ni. So ni after binding of the inhibitor to the allosteric site. So kesannya substrate can no longer bind to the active site. So substrate ni tak boleh nak bind to the active site. So when the substrate cannot bind means that less formation of enzyme substrate complex, okay, which also means lower the rate of reaction. So kita boleh nampak between competitive and non-competitive inhibitor, dia punya main effect dia adalah sama, iaitu untuk reduce the formation of VAC and then reduce the rate of reaction. Tetapi how do they did that is different. So kalau non-competitive adalah dengan dia bind kepada allosteric site yang akan causes change in the structure of the active site. Okay? Tetapi kalau competitive tadi, dia akan bind to the active site yang akan prevent formation of the enzyme substrate complex without changing the conformation structure of the active site. So itu untuk competitive. So make sure boleh differentiate antara the effect of competitive dan juga non-competitive inhibitor towards the enzyme activity. So the last subtopic is 4.5 classification of enzyme. So state the 6 classes of enzyme according to the International Union of Biochemistry and Molecular Biology IUBMB classification. So untuk ini awak hanya perlu state saja the 6 classes of enzyme, lepas tu example. So cara nak senang untuk ingat adalah awak guna akronim ni, mnemonic ni, iaitu hot lily. So the H stands for hydrolase, O is for oxidoreductase, T for transferase, L for ligase, I for isomerase, L for lyase. So ada 6 classes of enzyme according to the IUBMB. Okay, so guna ni untuk awak ingat hot lily. Okay, for hydrolase, so the function of the enzyme is to break bonds by using water. So hydro refers to water, ASE is the enzyme. So hydrolase is a class of enzyme that can break down anything by adding water to the chemical reaction. So basically any enzyme yang boleh buat hydrolysis itu adalah enzyme yang belong to the class hydrolase. So itu yang pertama. So yang opposite kepada hydrolase adalah lyase. So for lyase is basically similar to hydrolase, cuma it can break bonds without adding water. So maksudnya other than hydrolysis tapi also to break bonds, so itu adalah lyase. Okay, so hydrolase breaking bond, hydrolysis by adding water, tapi lyase adalah to break bonds without adding water. So itu je beza dia. So next is isomerase. Isomerase, kamu should be familiar with the term isomer. So it's an enzyme that involved in isomerization, reaction that change one isomer to another isomer. So yang kita tahu isomer adalah molecules yang the same chemical formula but different structure. So maksudnya arrangement of the molecule of the atoms in the molecule tu yang berbeza. Okay, sebab tu kita kata rearrangement. So rearrangement of the atoms to convert one isomer to another. Okay, so dia adalah isomerization. Itu reaction dia. Dia punya class of enzyme dia adalah isomerase. Next is transferase. So transferase is, as the name suggests, is to transfer functional group from one substance to another. Okay, so itu adalah transferase. Function dia untuk transfer functional group from one molecule to another molecule. Next is ligase. So ligase pun you should be familiar. So ligase adalah enzim yang akan tolong form covalent bonds between two molecules by using energy from ATP. Sebelum ni awak belajar DNA ligase yang akan tolong form phosphodiester bond. Lastly is oxidoreductase, a class of enzyme that can conduct oxidation and reduction reaction. So basically, enzyme yang akan buat redox reaction. Okay, again, you should be familiar with this reaction since awak dah belajar dalam masa sekolah menengah dulu dan juga semasa dalam subjek chemistry. So for the oxidoreductase, dia adalah enzyme yang akan buat both reaction simultaneously, which is oxidation and reaction. Okay. So for most of these classes of enzyme, kita akan tengok balik semasa chapter 5 nanti, iaitu cellular respiration. So that's it for the classes of enzyme. So that's it for chapter biocatalysis.